students. So I think you have seen the last videos, that is transcription and translation, and I hope that you are very clear with the transcription and translation. Okay, so there will be no doubt. So next we will talk about a very important concept, that is regulation of gene expression. So this regulation of gene expression we are going to study with the help of Operon concept. Okay, what is regulation of gene expression? First try to understand. Every metabolic reaction in any living organism, if you take a bacteria, if you take a fungi, if you take human beings, if you take mammals, any metabolic reaction, that will be under the control of set of genes. Okay, because you know that in our body some metabolic reaction requires enzymes, some metabolic reaction requires what we call as the hormones, and hormones are nothing but they are form of some of the proteins. Okay, so these enzymes, hormones which are made up of proteins, it means that they need what we call as some genes or a group of genes or set of genes. Okay, and all the genes regulating a metabolic reaction, that is called as operon, or the set of genes which regulating the metabolic reaction, that is called as what? Operon. So in that operon, which type of genes we are having? So we have a regulatory gene and we have a structural genes. Okay, regulatory gene, what it will do? It will mainly regulates the metabolic reaction, and structural genes are responsible for the production of what we call as the product what we want. For example, the enzymes, for example the proteins, for example particular amino acids. Okay, so a regulatory gene is regulating the metabolic reaction, and the structural genes are responsible for the production of product. Okay, so with the help of Lac Operon concept, which is studied in the E. coli cells, we are going to study the regulation of gene expression. So this Lac Operon concept was studied by two scientists called Jacob and Monod. So what was the concept? That concept we will study in the E. coli cells. So it is as follows. So these are the two scientists who have proposed what we call as the Lac Operon concept. So first of all, we'll study the structure of operon, what all it contains. Okay, the Lac Operon, which was studied in the E. coli cells, the structure of the Lac Operon is as follows. First you can see the promoter, second you can see the regulatory gene, next you can see one more promoter, next operator region, and three structural genes. So a regulatory gene codes for repressor protein. Regulatory gene codes for repressor protein, and three structural genes called LAC-Z, LAC-Y and LAC-A. These three genes codes for beta galactosidase, permease and trans-acetylase. And there is what? Operator region. Along with that, the promoters will also help in the metabolic reaction. So next we'll talk about the functions of the what we call as structural genes. So the structural genes, mainly three structural genes are there in this Lac Operon, that is Z gene, otherwise LAC-Z, codes for beta galactosidase, and beta galactosidase hydrolyzes lactose to glucose and galactose. Next, the gene Y codes for permease, and it increases the permeability of the cell, means the E. coli cell, to what we call as lactose. So E. coli cell's plasma membrane can easily allow the lactose to pass through it, and the lactose will enter into the cell with the help of enzyme permease. and the A gene codes for trans-acetylase. Scientists were not knowing about the function of the trans-acetylase, but anyhow it is present in the Lac Operon. So one or the other way it is helping in lactose metabolism. So next we'll talk about how this Lac Operon works. So Lac Operon works in two ways. One is a switched off operon and another is a switched on operon. So the switched off operon is called as negative regulation and switched on operon is called as positive regulation. Okay, why it is called as negative regulation and why it is called as positive regulation, that we will see one by one. So first of all we'll talk about the switched off operon, that is the regulation of gene expression through negative regulation. You know, here lactose is the inducer. Lactose is the inducer because we are talking about lactose metabolism. Okay, a metabolic activity in the presence of lactose and in the absence of lactose we are talking about. So first of all we'll talk about in the absence of inducer, that is inducer is lactose. You know, already we have studied the operon. In that operon we have a regulatory gene I. I refers to inhibitory gene. Inhibitory gene. Okay, this regulatory gene I normally produces the repressor mRNA, and repressor mRNA produces what we call as repressor protein. Okay, now in the absence of lactose, this repressor protein will go and bind to the operator region or the operator gene of the what we call as operon. And once it will bind to the operator gene of the operon, it will stop the movement of RNA polymerase. What is the function of RNA polymerase? You know, in prokaryotic transcription, a single enzyme codes for all type of RNAs. So here a single RNA polymerase will help in the transcription. So RNA polymerase movement will be stopped by the what we call as repressor protein. And once the RNA polymerase will stop, there will be no synthesis of mRNA. If there is no synthesis of mRNA, automatically there will be no synthesis of proteins. It means there will be no synthesis of what we call as enzyme beta galactosidase, permease and trans-acetylase. Okay, and this type of regulation is called as negative regulation and the operon is called as switched off operon. This is what we have studied in the absence of inducer, that is in the absence of lactose. Next we'll talk about in the presence of lactose. In the presence of lactose, usually again what the regulatory gene I produces the repressor mRNA produces repressor protein, but now the inducer lactose is present. Inducer lactose is present. The inducer lactose binds to the repressor protein. When inducer binds to the repressor protein cannot bind to the operator gene of the operon, and when it will not bind to the operator gene, what will happen? It cannot stop the movement of what we call as the RNA polymerase, and RNA polymerase will be active, and RNA polymerase will be active. Then what will happen? The structural genes will be switched on, and the structural genes with the help of RNA polymerase will produces the mRNA, and after that it will produces what we call as the enzyme beta galactosidase, permease, trans-acetylase with the help of translation. Okay, so beta galactosidase, then what? It hydrolyzes the lactose into galactose and glucose. Okay, and once if lactose is completely utilized by the E. coli cells, Once the lactose is completely utilized by the E. coli cells, what will happen then? The lactose cannot stop the repressor protein. Then the repressor protein again go and bind to the operator region or operator gene, and again the movement of RNA polymerase will be stopped, and again what we called as the operon will be converted into switched off operon, and what? Again there will be no synthesis of mRNA, and there will be no synthesis of beta galactosidase, permease and transacetylase, because there is no lactose, so there is no need of beta galactosidase. Understood? So in the absence of inducer, it is called negative regulation, and in the presence of inducer, it is called positive regulation. Okay? So in the absence of inducer, it is called as what? Negative regulation. There what happens? Repressor protein stops the activity of structural genes. Here, in the presence of inducer, okay, inducer binds to repressor protein and the structural genes will be active and they will produce what? The enzymes. This is called positive, what we called as regulation. So by this you can easily understand what we call as the regulation of gene expression. But the bacteria have to carry out its life activity. So that's why there are some metabolic reactions. There are some metabolic reactions can be carried out continuously. Can be carried out what? Continuously. And because of that, these genes cannot undergo what we call as switched off or switched on system. Okay, the process should be continuous. For example, take example of glycolysis. Okay, glycolysis is a metabolic reaction where it will provide what we call as energy. So it has to be processed continuously. So that's why in such type of metabolic processes, genes will be continuously active. They will be not undergoing switched off or switched on concept, and thus this type of what we call as genes are called as housekeeping genes, and the processes or the metabolic reactions are called as housekeeping metabolic reactions. Okay, so I hope students you have understood the regulation of gene expression. Yes, if you are having any doubt, again you are free to ask in the online classes, and definitely the Srinagar Public School and the teachers definitely will help you to sort out your problems. Okay, and revise the things, go through this video, understand the concept, and go through the NCERT textbook also. If any doubt, again you are free to ask. Thank you, students.